Okay, so we're going to review by going through the notes, and then we'll do a few problems in the picture packet. So basic chem, we had said this is a good review of middle school. The Regents and the test isn't going to have a ton of this, but you need to know it to move on with the biochemistry content. We talked about chemical formulas. You write the letter with a corresponding number. Structural formulas, we talked about the line shows the bond. Remember, carbohydrates are always in the shape of a ring. We talked about reactants present before the reaction starts. We talked about the arrow is yields, products which are produced by the reaction. We got into the pH scale. This was super important. You want to study this definitely for the test. The Regents will tend to have maybe 9 to 10 points on it that have to do with pH. So remember, the pH scale goes from 0 to 14. 0 to 6 is an acid. Remember, a strong acid would be closer to 0, 0, 1, 2. Weak acid is 6. Neutral is 7. A base is 8 to 14. A strong base would be like 13, 14. A weak base like 8 or 9. We talked about hydrogen ions being part of the chemical makeup of acids, and hydroxyl ions go with the base, put a hydrogen ion together and a hydroxyl ion. So an H, an H, and an O, what do you get? Water, that it's neutral. Okay, this is the hard stuff. This is what you primarily want to be studying, the biochemistry. So if you're organic, what did we say you have? Carbon, hydrogen, and oxygen. And then basically the rest of this unit are about the main 4 organic compounds: carbohydrates, lipids, proteins, nucleic acids. You want to know everything about these, inside and out. Okay, water, we said, is the most important inorganic, so it doesn't have carbon, hydrogen, and oxygen in our body. It's the most important inorganic compound in our body. Along with salts, acids, and bases are also inorganic. They're in our body as well. The 2 reactions, you only have to know 2 reactions. So dehydration, synthesis, you start with 2. See, there's a plus sign. Those are your reactants. And you end with 1. Okay? Dehydration, synthesis, start with 2, end with 1. Hydrolysis, start with 1, end with 2. So now you see 2 here for products. Hydrolysis is to break. In dehydration synthesis, we take the water out so the bond can form. In hydrolysis, water gets added in. Remember, we talked about the molecules vibrating until they're stable. They wanted to bond with the H and the OH. Again, the water piece is higher level. You're not going to have to explain that on the Regents type questions, but in the big picture, I just wanted you to understand that. Next year, you'll talk more about that. All right, here's our carbs page. How do you feel about carbs? If you don't feel comfortable with this, this is the page you want to spend some more time on. Carbs, sugars are another name, right? They are fruits and vegetables. They have carbon, hydrogen, and oxygen in a 2-to-1 ratio. They are our main source of energy. Carbs are used to make ATP. That's what the body runs on. They always end in ose. We talked about that. They're always in the shape of a ring. Saccharide is another name for carbs. Monosaccharides 1, disaccharides 2. Poly would be many. Polymer is another way to say many, like a chain. So you have a big plate of pasta, we said, and then you sit on the couch after dinner. Okay, you did not burn up all that sugar. So we store it, animals store it as glycogen in our liver and muscles. Plants store excess carbs as starch. We talked about like a potato. They can also store it in the outer organelle, the cell wall around the edge of the plant. That is stored as cellulose or fiber. And then we talked about insects. They store it as chitin. That they tend not to ask that on the Regents, but I wanted to go over it. And you definitely need to know that chemical formula, C6H12O6. Lipids. So self-assess. See how you feel about lipids. You might need to spend more time with lipids. Again, the same 3 elements if you're organic: carbon, hydrogen, and oxygen. But now you're not in a 2-to-1 ratio. Okay, your quiz last week had a question with those 4 chemical formulas, so you need to know that. Lipids, most kids remember fats, and they kind of forget oils and waxes. Okay, don't forget, lipids also, oils and waxes compose lipids. The function is the cell membrane. It forms the outer edge we talked about. It provides our backup energy, okay? And it cushions the body when we fall, and it helps to keep us warm. It insulates us. And then we talked about the building blocks of lipids. Remember the building blocks of carbs? Simple sugars. The building blocks of lipids, I sang it to you, remember? 3 fatty acids and a glycerol. So these are your 3 fatty acids. They can change. There's different types of fatty acids, and this is what glycerol looks like. Yields, this whole thing is a lipid, and remember, you always have to write the water. This time we have 3 waters, 1, 2, 3, from where we take it out, the H, and the O, so the bonds can form. We talked about saturated fat and unsaturated. Remember, saturated has cholesterol, and it can build up in our arteries if we have a diet high in saturated fat. We said which tends to come from animals, not always. And in America, a lot of people have a high saturated fat diet. So be mindful of that when you're making food choices. Unsaturated fat's a better choice. We said like nuts, seeds. Okay, we talked about avocado, fish, have both. Okay, proteins. This may be where you need to spend some time, okay? Carbs and lipids, we've gone over a ton. The newer stuff, protein, nucleic acids, maybe that's where you need to study more. So protein, we drew an N should be flashing at you. Protein ends in N, that was a little trick. So now we finally see a new element, nitrogen, carbon, hydrogen, and oxygen. But remember proteins, they have a ton of jobs. Repair, maintenance of cells and tissues, makes pigments, hormones, muscle tissue. Antibodies, remember they're in the immune system made by white blood cells. They're a big part of our defense against pathogens. And then enzymes. Enzymes, they love to ask about. We covered a whole part about enzymes at the end of the unit. What are the building blocks of proteins? The 20 different kinds of amino acids. Okay? At the college, if you take college biology, you'll have to know them all. But for Regents biology level, you just need to know the general structure. So we talked about this. We drew it in our notes, actually. These are called end arrangements or side groups. You're going to need to know the amino group is the N to the 2 H's. The carboxyl group was the C double bonded O. I drew the O there because it's hard to see. O to the OH. You drew that in your notes. The hydrogen. And then R, remember R is not on the periodic table of elements. It acts as a variable, just like in math. This is like X, Y, or Z. Okay, sometimes this is 3 carbons long. It might be 5 carbons long. This is how the 20 different amino acids differ by the variable group. Okay, just be mindful. We talked about molecules turning and bending. So if you see on the test that the R group is up here and the H is down, it's okay. They just turned. Okay, these are 3D molecules. The C double bonded O might be up high, and then the OH might be down here. Just be flexible when you're looking at molecules. Now, we named the bond when the building blocks came together. So, when 2 amino acids come together, the bond is called a peptide bond. We did not name the bond for lipids or carbs. They just name it for amino acids. So take a look at it. Here's 2 amino acids, and how do I know that? I see that amino group, the carboxyl group, and the R group. I see the plus sign, so I know these are reactants. There's the arrow, yields. There's a protein. Okay, we highlighted the peptide bond. That's the name of that bond. And now I can call that, remember, biology's vocabulary rich. I can call this a protein, but I can go and throw in the word dipeptide, just like we did disaccharide when 2 rings came together. We talked about the prefixes. So di is 2 amino acids. Poly would be more than 2. All right, so that was protein. You may need to re-look while you're studying at the structure of an amino acid. I have quite a bit of that, a couple questions about it on the test. All right, my lights just went out. All right, moving on. Nucleic acids. We're going to do that whole unit in March, the genetics unit, and get really in-depth into nucleic acids. It's so interesting. It's where the cutting-edge current biology information is at. I love learning about DNA and RNA and teaching you guys. So hopefully we're back to school by then. But nucleic acids, there's 2 types. They store and communicate genetic information. We talked about the two types. Here's a picture of them. There's DNA and RNA. So DNA, deoxyribonucleic acid, we practice saying that. Like I said, you don't, on the Regents, you can just write DNA, but you got to be able to recognize it. Deoxyribonucleic acid. The double helix just means there's two strands. RNA, ribonucleic acid. That's the single-stranded one. What did we say about DNA? Where do we find it? Only in the nucleus. Remember, it's just so important. All the steps of a ladder are the genetic code. They code, genes get turned on, and code for everything in your body. It's so important it stays only in the nucleus. RNA is the helper that goes into the nucleus. All right? And they both have the building blocks, nucleotides. Remember, I put it up here, but it's the same thing for both. Nucleotides. You need to know what the nucleotide consists of. A phosphate, a sugar, and a base. Okay, so those were the 4 organic compounds. When you are studying, we talked about this. Just like I do in class, I model, I mix them all up. So have someone at home mix them up, ask you questions from all 4 of them. This isn't really a flashcard type of study skill. Now in the picture packet, we put all 4 on one page. So for studying, you could, let me grab a sheet of paper. You could do this again. Okay, put all 4, make a foldable, and study from the foldable if you prefer to have them all together. All right, you need to be studying for this big test. Okay, you could do it that way. All right, let's just finish off the notes. We'll go over enzymes. Now, this is an example of a protein. I told you we're going to chat just about enzymes. So we spent we did that big Jello lab that we did was all about enzymes. So enzymes are protein substances, okay, necessary for chemical reactions. They are an example of a catalyst. I said next year in chem you'll talk more about different types of catalysts. But for now, enzymes require coenzymes. We chatted about coenzymes. Do you remember coenzymes were vitamins? An enzyme is a substance necessary for chemical change. We said they do not change, though. So remember in the diagram we'll review in a second, you always know the enzyme because it never changes. They can be used over and over again. They're made by cells. You can be used within a cell or like within the stomach, the small intestine. What do they act on? Remember, they act on the substrate. So we talked about what you had for breakfast, and we talked about the enzymes that break down that food that you had for breakfast. We said they always end in ase. So let's review. Maltose, what's the enzyme? Maltase. Protein, protease. Lipid, lipase. What about starch? Remember, you want to say starchase, but that's not what it is. It's salivary amylase. Amylase. That's the one that I said, okay, you eat this big plate of pasta or bread or something full of starch. It's the only enzyme that we add right to the food in the mouth. Remember that? Higher level question. Why? We talked about it being, let me see if I still have a picture. All the long chains, right? Because we want to slowly start breaking it down. So we want to get that enzyme right on it right at the beginning to start the chemical reaction of hydrolysis. We talked about where the enzyme and substrate meet is called the active site. Okay, we took our time. We drew this in our notes. We made a really nice diagram. They love photos like this with enzymes. All right, so where the enzyme and substrate meet is the active site. We call this the enzyme substrate complex when they are together. And then in this case, the substrates they get broken down to A and B. So I asked you a big higher level question. I'm like, okay, what is the name of this reaction? Okay, most of you knew it's hydrolysis. The food is here. So whatever you had for breakfast or lunch, you start with that, and you're seeing it 2 end products. Now, on the Regents this year, they might start with this, okay, the A and B, and then end with the substrate together. Then you know it's dehydration synthesis. All right, so when you look at this, remember the enzyme doesn't change, the substrate does. They fit together like a lock and key. We talked about how they fit right together. So if I put a protease on maltose, it's not going to fit. It can't fit together. All right, the active site doesn't match. It's very specific to the substrate that it's meant to lock into. All right, the very last thing, temperature and pH. So we talked about factors influencing enzyme action, temperature and pH. So the rate of enzyme action varies, and only small amounts are needed to change large amounts. Remember, it can be used over and over again. So temperature. Temperature increases, enzyme rate usually increases. So here's a graph. As temperature increases, enzyme rate is increasing. So faster motion of molecules, they start to hit more, they're touching the active site more. But what happens here? At high temperatures, enzymes begin to break down or denature. We said underline it, highlight it, put a little asterisk. This is what the whole Jello lab was about. Remember the canned pineapple solidified. Why didn't bromelain digest that gelatin? Because remember in the canning process we talked about you boil. So in that factory, the bromelain was denatured. At high temperatures, enzymes begin to break down. If the shape changes, they break down and the shape changes, and now this becomes like straight across, it's not going to fit. So when you have a fever, we chatted about this. Why is it so important to bring that fever down? You don't want your enzymes to denature. If this starts to go straight across, your organs can't do their chemical reactions. If in high fevers, high temps, this is not maintaining homeostasis in the body. And then those organs will slowly start to shut down. All right? So temperature is super important. Also pH, many enzymes work best at a specific pH. But there's none that you have to memorize. They have to give you a graph. They have to give you the information. So here's a couple of Regents questions. This is pepsin. So what pH does pepsin is optimal for pepsin activity? So you would say 2, right, at the top. What is it for trypsin? Then you would say 8. All right, let's just review a few by the picture packet. This, you don't have to spend a ton of time reviewing. This was that general chem stuff from 8th grade. I definitely spend time studying here, okay, dividing them up, having them all on one. Now, we did this in class. This was a great review, so when you are studying, here's another way to study. This was about being organic. This is carbs. This is lipids, proteins, nucleic acids. Put a piece of paper here. I would do this again. If this is coming easy, then you're really understanding carbs, lipids, proteins, nucleic acids. All right, so let's just do a few questions. Based on your answers on the equation, which represents a biochemical reaction on your knowledge of biology, to which class of compounds do the products of this equation belong? So as soon as I see this, it should be a light flashing, rings, 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 carbohydrates. The rate of the enzyme action of the enzyme protease is affected by, we just went over it, right? Temperature, pH, and obviously how much the concentration you have. Okay, so this is the only one. So see how this says temperature, pH, but it says carbohydrate concentration? They're testing you if you know protease is protein. Okay, so that's why it's 2. Compound X increases the rate of the reaction below. Compound X is most likely an enzyme. They always put enzymes on the arrow. Just assume every reaction there's enzymes present on the arrow. Which statement concerning the water molecule in this equation is true? Okay, so water's present at the beginning, okay? And this is a higher level question, like I said. You're not going to see this on your test. So water's present in the beginning. And then this is hydrolysis, right? So when it breaks, it ends up becoming part of it. So it splits and becomes part of the product molecule. 5, gastric protease operates most efficiently in the presence of hydrochloric acid. Which pH range would be optimal for gastric protease? So first they're testing you whether you know an acid. So an acid we know is 0 to 6. Now they're bringing up gastric protease. All right, so they're really just testing you on which numbers here would work. So number 1, 1 to 3. Fatty acids and amino acids both contain, all acids contain carboxyl groups. We talked about that in the, when we looked over the carboxyl group on the amino acid. All right, so you want to definitely go through and study, use the notes, the picture packet, practice questions, do your Castle Learning assignment. You can go back and redo the Kahoot we played. Use all of these tools to help you study.